Greetings, good people. We meet once again to look at digestion in ruminants. Earlier on, we were just looking at the alimentary canal of ruminant animals. But what we want to do now, we want to go into details and look at the digestion that takes place in a ruminant animal. This is what we have to focus on when we look at this one. The first thing, we must be able to define these three terms. We must be able to define rumination, define regurgitation, and also define peristalsis. We can sometimes get these terms in section A, especially in 1.3 or else in 1.4, but mostly they are in 1.3. So we will look at the definitions of these three terms. We have to look at the rumen microbes. We must be able to describe the rumen microbes as single cell organisms found in the reticulo-rumen. We have three of them, so we will try to look at those three and how can we identify them. Sometimes they also give you a question based on this one in 1.3 as a term. They say this is a single cell organism that has this duration of life in the reticulo-rumen, but we will look at them later. We have to now classify the different types of rumen microbes. We have to classify the different types of rumen microbes. It's what we will do. Again, we will then categorize bacteria into different types of bacteria or different organisms found in the rumen. We call them rumen bacteria. And when we categorize them based on their function. So we will look at, we have this type of bacteria which is cellulolytic, and then we'll look at, say, what is the function of cellulolytic, what is the function of proteolytic, and what is the function of amylolytic. So the names that are given to these rumen bacteria are based on their functions. We will look at this one as we move along. We have to talk about the functions of the rumen microbes. We have three of them. If we don't confuse the cellulolytic and the other three microbes, we have to look at their functions. What are their functions? There are four functions that we have to talk about. The last part of this gathering that we will check, we have to talk about the requirements for the proper functioning of the rumen microbes or the rumen microflora. So that's what we will do as we move along here. Let's try to go step by step in understanding what we have to check here. When we talk about rumination, good people, remember we said the difference between a ruminant and a ruminant and a non-ruminant is that a ruminant animal can regurgitate. So when we talk about the rumination process, we say is when food is chewed, swallowed, brought back to the mouth, and chewed again. So when we talk about rumination, we say this is the process whereby the food will be chewed a little bit, swallowed by the animal, and when it is swallowed by the animal, it will go to the reticulo-rumen or to the rumen and reticulum. Remember, the combination of a rumen and reticulum, we said, is reticulo-rumen. So it will go to the reticulo-rumen, and then it has to go back to the mouth. We will check why the food has to go back to the mouth, and then it has to come back again to the stomach, and it will land into the omasum. So by this definition, we can understand the stages of what? Of rumination. The food is swallowed for the first time. It goes back to the mouth for proper chewing, and then it comes back to the stomach again. But then we will get to those stages later on. Let us look at this other term, because I said we have to look at these three terms. We also have peristalsis. We talked about peristalsis when we were looking at the function of esophagus. when we look at the digestive system of what? Of a cow. When we talk about peristalsis, in a simpler way you can say is the movement of food along the alimentary canal. But what we said here, we said is the process that pushes food through the alimentary canal by the relaxation and contraction of the longitudinal and circular muscles in the walls of the alimentary canal. If you try to recall very well, we once had this diagram whereby we said that this was food that was going downwards, food that was going downward. And we call this process the movement of food along the alimentary canal. We give it a name and say it's what? It's peristalsis. Okay, fine. We have one more term to discuss, good people. What is the name of that term? Regurgitation. You can sometimes get this term in your 1.3. When we talk about regurgitation, it's simple. It's the process of bringing back the food into the mouth for what? For proper chewing or for remastication. We say it's remastication because the food is chewed again. Mastication is chewing. So when we say remastication, the food will be chewed again. And we sometimes call it chewing the cud. So these are the terms that you have. Rumination, the food is swallowed, is chewed, swallowed, and then brought back to the mouth for proper chewing. And then again, it will come back to the stomach. Peristalsis, relaxation and contraction of the longitudinal and circular muscles of the alimentary canal with a purpose of pushing the food down the alimentary canal. Regurgitation, process of bringing back the food into the mouth for proper chewing, which is chewing the cud. Then, good people, let us get into the stage, or let us get into the process of what? Of rumination. When we look at this process of rumination, it is made up of 3 stages. It is made up of 3 stages. What do we mean by these? What are these 3 stages? Let's understand it in a simpler way. We said that the animal will swallow the food, and then the food has to go back to the mouth for proper chewing. It has to come back to the stomach again. There are 3 stages. 1, the food goes into the reticulo-rumen. 2, the food goes back to the mouth. 3, the food comes back to the stomach, but at this point of time, it lands into the omasum. Let's try to look at these stages. We have peristalsis. This is the first stage, whereby the food lands or the food moves through the or along the esophagus. It goes into the reticulo-rumen. That is stage number 1. What is stage number 2? We can call it reverse peristalsis or retroperistalsis. When the food is going back, is reversing, is going back to the mouth for proper chewing, we call it reverse peristalsis. Sometimes you can call it regurgitation, whereby we said is the process of bringing the food back to the mouth. Then we have the last stage. The last stage, we give it a name and say is re-swallowing. Now the food is swallowed for the second time. So we call it what? Re-swallowing. 3 stages. It enters for the first time, peristalsis. It goes back to the mouth. So it is reversing, retro or reverse peristalsis. It comes back to the stomach for the second time. We call it re-swallowing. Let's take note of this part before we move on. When the food enters for the first time, it will land into the rumen, and then it will go to the reticulum. Then the reticulum will push the food back to the mouth for what? For proper chewing. When it enters for the second time through the re-swallowing process or stage, it will now enter the stomach called omasum. Sometimes you can get this question whereby they ask you, which chamber where the stomach lands for the second time? Through the reswallowing process, you will say it's omasum. So that's what I want you to understand here. But then, good people, let us look at it in this diagram that we have. We have these three. We have this diagram. Diagram A, we have, in this diagram, we have letter A. This is your letter A. This is your letter A. The food enters the stomach for the first time. Which stomach where it lands? It is what? It is the rumen. We said the process or the stage whereby the food enters the stomach or one of the chambers for the first time, we call it peristalsis. Once it enters, then the food, good people, I don't know whether you will be able to see this one. Then the food, good people, has to go back to the mouth. Has to go back to the mouth. And which stomach pushes the food back to the mouth? It is the stomach that we call reticulum. It is the stomach that we call reticulum. Then it goes back to the mouth. Which process whereby the food goes back to the mouth, we call it reverse peristalsis, because we said when it enters for the first time, it's peristalsis. But when it has to go back for the second time, when it's to go back to the mouth, we call it reverse peristalsis. Okay, good people. Now, the food has to come back. The food has to come back to the stomach or to the chambers. When the food comes back, it will land in this stomach that we call omasum. What is the name of this process or the name of this stage? We call it what? We call it re-swallowing. 3 stages. It enters for the first time, peristalsis. It goes back with our green arrow there, reverse peristalsis. It comes back with our red arrows there. It's what? It's re-swallowing. That's what I want you to understand from this diagram, good people. They can give you this diagram. When they give you this diagram, maybe one question that you may get, they can say, classify the type of animal with such alimentary canal. There are 4 chambers, remember. You will say it's a ruminant. Then you provide your reason. It's because the animal or their stomachs are 4. It's made up of 4 chambers. Then they will ask you the other questions. But let's continue. We will get to that part later. This is what we have. We said our A, it's peristalsis. Our B, it's reverse peristalsis. Then our C, it's re-swallowing. Let's ask ourselves a question now, my good people. Why the food has to go back to the mouth? That's the question that you can ask yourself. Why the food has to go back to the mouth? Importance or reasons of regurgitation. You will get this question. Why food is regurgitated? Or why stage B, which is regurgitation, is important? You can have a number of reasons there, but make sure that you are able to recall at least 3. 1, we say food broken down mechanically into finer particles. For the food to be broken down into finer particles, hence we say it's proper chewing. 2, you can say it increases surface area of food. It increases the surface area of food. You can also talk about that it improves the mixing of food, with what? With the saliva. You can also say it's for the formation of what? Formation of bolus. That's what you can check. Then the last one you can talk about, it stimulates the secretion of saliva to maintain rumen pH levels. Please make sure that you are able to recall at least 3 of them. Increases the surface area, improves the mixing of food, or the formation of what? Of bolus. Okay, let's get to this important part, good people. Reticulo-rumen digestion. When we look at the reticulo-rumen digestion, we have 3 microorganisms that we get, 3 microflora that we get here. Remember, we said what takes place in the rumen and reticulum, it's microbial digestion. It's microbial digestion. So we're trying to look at it now whereby we say it's reticulo-rumen digestion. Now we will answer the question that says, how is, remember I talked about this one, how is a ruminant adapted to digest what? Cellulose. Okay, fine. What I want you to notice that we have 3 microorganisms that we have here. One, we have a microorganism that is called bacteria. These microorganisms that are called bacteria are the ones that are more with a high population in the reticulo-rumen. Take note of this part. They make about 60 to 90% of mass in the reticulo-rumen. Their generation interval is about 20 minutes. It's about 20 minutes. Then you can say they are more. Their numbers in per milliliters in the stomach, about 25 to 80 billion. You also talk about the protozoa. The protozoa, if you look at the mass of the protozoa in the reticulo-rumen, it's about 10 to 40. In terms of their survival generation interval, it's 8 to 36 hours. 8 to 36 hours. When you look at their numbers per milliliters, you can talk about 200 to 500,000. We have the last one, fungi. Fungi, you in terms of the mass, it's about 5 to 10% of mass. Their survival, it's about 24 hours. It's about 24 hours. Then there are very few in the stomach. So the way I try to list them, the bacteria are the ones that are more in the reticulo-rumen, the protozoa, they are meager, then the fungi, they are less, they are minimal. So, but most of the time they used to say least. Then there can be those times whereby they say a single cell organism found in the reticulo-rumen that has a mass of 5 to 10%. You will say it's fungi. A single cell organism that has a mass of 10 to 40%, you will say it's protozoa. A single cell organism that has a mass of 60 to 90%, you will say it's bacteria. So make sure that you highlighted this part that I've just said to you. Okay, fine, good people. This is where I want me and you to go. We will come back to the functions of this. We say that there are 3. We have the bacteria, protozoa, and fungi. When we look at this, good people, let us be specific now and talk about the rumen bacteria. The population of bacteria that we get in the rumen, species of bacteria that we get in the rumen, species of rumen bacteria. This is what I want you to understand. We remember we said we will focus on the first 3, which is cellulolytic, amylolytic, and proteolytic. But then I will just highlight the other 3 more. But those are the ones that you can get, that you can cram more, which mostly appear in the exam. Okay, fine. This is what I want you to note. I want you to know the name of the organism or the name of the bacteria. From then, know the function. For you good people to master the name and the function, or to master the function, it must be clear to you that these organisms or these bacteria are named based on their functions. If you can understand their name, it will be easier for you to understand their function. Okay, fine. We have the first, which we call cellulolytic organisms. Let's try to understand this one. When we look at cellulolytic, this is what I want you to understand. Now we are trying to understand their function. A cellulolytic organism, the word cellulo is taken from the feed, which is cellulose. The word cellulo is taken from the feed, which is cellulose. Okay, those are your refuges. Okay, fine, we're not there. The word lytic, we can make it simple and say it comes from the term called hydrolysis. It comes from the term called hydrolysis. What do we mean by hydrolysis? We are simply talking about breaking down. If we talk about breaking down, it simply means we talk about digest. I hope you follow the argument. Let's repeat it again. The word cellulo comes from cellulose. The word lytic, we can agree that it comes from hydrolysis. If we try to explain hydrolysis, when we scratch the surface, we can say hydrolysis, it means breaking down or digest. That means, good people, that means the cellulolytic organisms or cellulolytic bacteria, these are bacteria that digest what? Cellulose. Hence, we call them cellulose digesting organisms. I hope you are clear on that one. I hope you are clear on that one. I will come back when we summarize it. The second rumen bacteria, amylolytic. Amylolytic. Again, good people. Again, the word amylo is taken from an enzyme. This enzyme, we give it a name and say it's amylase. The function of the amylase, in a simpler way, is to digest. What do they digest? They digest starch. I'm done. Amylo, it's an enzyme. The function of the enzyme is to digest starch. Lytic, we said hydrolysis, meaning breaking down or digesting. What does that mean, good people? It means the amylolytic organism or amylolytic bacteria. This is a bacteria that digests what? Starch. You are done. You get your mark. Thank you. Let's look at this one. Proteolytic. Proteolytic. Lytic, we are fine with lytic. Hydrolysis or breaking down, we are safe with lytic. Let's talk about the proteo. The word proteo, the word proteo is taken from proteins. If we say it's taken from proteins, what does it mean? Let's bear in mind that our lytic, it simply means what? Digest. What does it mean? It means the proteolytic organisms, these are organisms that digest what? Proteins. These are organisms that digest what? Proteins. So, good people, we say, if I can try to close it here, cellulolytic, cellulo from cellulose. So what does it mean? These are organisms that digest cellulose. Amylolytic is taken from amylase. What is an amylase? It is an enzyme that will digest what? Starch. So what does it mean? It means amylolytic bacteria, it's a bacteria that digests starch. Proteo is taken from proteins. So it means this is an enzyme, or this is an organism that digests what? That digests proteins. Then you have the others, lipolytic. Lipo, we can say, is taken from lipids. So it means this is an enzyme that digests lipase or lipids. Hemicellulolytic, it digests hemicellulose. Then you also have fibrolytic, it digests what? It digests what? Fiber. This is what I want you to note, but make sure that you focus more on the first three. The others can also come in the exam, but mostly they like to examine you based on the first three. Okay, fine, good people. Let's come here. This is what I want you to understand here. The number of these microorganisms, whether it's cellulolytic, it's amylolytic, it's proteolytic, or else it's lipolytic, hemicellular, or else fibro. The number of these organisms in the stomach, they depend on the composition of the feed that the animal consumes. What do I mean by this? We simply mean that what will depend on the population of these organisms will be based on which type of feed the animal has ingested. Okay, fine. Let's say the animal eats cellulose. If the animal eats more cellulose, what does it mean? It means the population of cellulolytic bacteria in the rumen will increase. If it eats cellulose, it means the bacteria that digest cellulose will be more in the stomach, which is cellulolytic. If it eats more feed that contains proteins, it means the bacteria that digest proteins will be more in the stomach. Which are those bacteria? Proteolytic. Okay, fine. This is what we have. Amylolytic bacteria, they say, will increase in the stomach if the animal feeds on ration with a large quantity of starch. So remember we said amylolytic, they digest starch. So once the animal eat feed with more starch, the number of amylolytic bacteria will be more or will increase in the stomach. I'm done. Cellulolytic will increase if the animal feeds on a ration that has high roughages. So you give the animal more roughages, the number of cellulolytic bacteria or the number of bacteria that digest roughages or cellulose will be more in the stomach. The last one, proteolytic bacteria, will increase if the animal feeds on a ration that is rich in what? In proteins. That's what you have to understand it. So they can question you in a different way, whereby they say, okay, fine, the grass there contains more proteins, the grass contains more roughages, the grass contains more starch. Then they ask you which bacteria will be more in the stomach. So just understand the bacteria that will digest the feed that is more in that grass or else in that ration. Okay, fine. We're about to come to an end, but let's look at the functions of the microorganisms. Microorganisms have 4 functions. I will not spend more time here. The first function of the microorganism, now we go back to the 3 microorganisms, the bacteria, rumen, and fungi. Don't confuse these functions with the 3 types, with the cellulolytic, proteolytic, and amylolytic. Please don't confuse the 2 groups. One function is that they synthesize amino acids. You will get this question whereby they ask you, which animal is able to synthesize amino acids? You will say it's a ruminant. Why? Because they are microorganisms that are able to synthesize amino acids. We will explain later how how it does that, whereby it uses the ammonia to synthesize amino acids. Okay, fine. We also have second function whereby we say it synthesizes vitamin B complex and vitamin K. So it synthesizes the vitamin B complex and vitamin K using a cobalt. We also say it will hydrolyze proteins or digest proteins. It's what we have just checked now. We have the last one whereby you say it digests cellulose, which is the microbial fermentation that we talked about. So we have 4 functions. Make sure that you know all of them. Never forget even a single one. So we say synthesize amino acids, synthesize vitamin B complex and vitamin K. It also hydrolyzes proteins and it digests what? Cellulose. Okay, fine. Let me go deeper with the digestion of cellulose, good people. Let's talk about the digestion of cellulose. When cellulose are digested, they will be digested into different products. The first thing that I want you to notice that the microorganisms will secrete an enzyme which is called cellulase. So instead of cellulose, you just replace the O with A. So the microorganisms will secrete an enzyme which is called cellulase. This enzyme will digest the microorganisms. So will digest the cellulose. They will digest the cellulose into what? They will digest the cellulose into 2 fermentation gases and also into 3 volatile fatty acids. It's very important for you to know the name of those fermentation gases and the name of those volatile fatty acids. So we are saying it will digest the cellulose into what? 1, into 2 fermentation gases. What are those fermentation gases? Methane and carbon dioxide. They like to ask it in section A, your multiple choice. Into the methane and carbon dioxide. Then they will also digest the cellulose into 3 volatile fatty acids. What are those volatile fatty acids? It's your acetic, your propionic, together with what? With the butyric. Make sure that you know all of them. So we are saying what will assist the digestion of cellulose? It's an enzyme. So these are a microorganism that secrete an enzyme called cellulase. They will digest the cellulose into what? 2 fermentation gases. What are the name of those fermentation gases? Carbon dioxide together with what? With methane. And then they will digest them into 3 volatile fatty acids. What are the name of those volatile fatty acids? Acetic, propionic, and butyric. Okay, fine. Ask yourself a question, good people. What will happen to these fermentation gases and what will happen to these volatile fatty acids? That's what we must ask ourselves now. Yes, we know that we have these 3 gases, these 2 gases and 3 acids. What happens to them? What happens to the fermentation gases? These fermentation gases, good people, they must be removed by the animal through a certain process, because failure of the animal to remove the fermentation gases, it will result to a disorder that we call what? Bloating. Let's look at it. The fermentation gases are removed. They are removed through a process that is called eructation, or you can just call it belching. They will ask you this question in section A 1.3. The process of removing the fermentation gases from the stomach, you will say belching. Okay, fine. Let us say these gases are not removed. What will happen to the animal? The animal, if the animal cannot remove the gases, they will suffer from what we call a bloating. That's what you can understand here. The name of the process of removing the gases is what? It's belching. Let's say the animal fails to remove the gases. They will suffer from what? From bloating. Okay, fine. What about the gases? What will happen to the gases? Sorry, to the volatile fatty acids. These volatile fatty acids, good people, they must be absorbed. If you remember very well, when we talked about the rumen, we said in the rumen there are finger-like projections. Try to recall. What is the name of those finger-like projections? We say they are what? They are papillae. And I highlighted the functions of the papillae. There were two. Now let us look one of the functions. We are saying the volatile fatty acid are absorbed in the rumen by what? By the papillae. They are absorbed in the rumen by what? By the papillae. That's what you can understand here. You will get this one in, so they can ask you that what takes place in the rumen. You will say absorption of the gases. Okay, fine, good people. Let's close it by looking at this one. Requirements of rumen microflora or microorganisms. What is the condition that is required for proper functioning of these microorganisms? One, you will say they need a free oxygen environment or absence of oxygen, which is anaerobic environment. The temperature must be warm. Then we can say plus or minus 38 to 42 degrees Celsius. We can also talk about the moist or wet environment. That's what you can talk about. If you can cram these three or these four: absence of oxygen, warm environment or warm temperature, wet or moist environment, and then you can say slightly acidic pH. That's what you can get. Then we can continue and say sufficient nutrients, which is regular feed intake. You can talk about easily digestible carbohydrates, sufficient mineral nutrients, sufficient nitrogen, or a proper excretion or proper removal of waste product. But if you can cram the first three that are on our right or our left, sorry, we can be safe. This is what you have to understand, good people. If you get if it happens that you get an alimentary canal of a cow, just know that one of these or two or three questions from this lesson, you will get a question from this lesson. What you must know, especially the digestion, you must know the microorganisms that are involved there. You must know the function of the microorganisms, the requirements of the microorganism. If you talk about the rumen bacteria, you must know how are they different based on their functions. We still gonna talk about adaptation of the ruminant in terms of digesting cellulose. But just to brief you, we will say a ruminant is able to digest cellulose. One, because they have, take note of this one. One, because they have a reticulo-rumen that contains microorganisms that are able to digest cellulose. They have a reticulo-rumen that contains microorganisms that are able to digest what? Cellulose. That is the adaptation. But then we will have a lesson whereby we look at it thoroughly. It will be based only on the adaptations of some of the parts of the alimentary canals. This is what we have. I'm sorry for taking too long in this lesson, but I wanted to be as low as I can for all of us to understand it. More videos will be uploaded. Make sure that you hit the notification button and also subscribe for more videos, good people. Thank you.